Hello guys, today we are going to learn about lipids. So a lipid is one of the four biomolecules, right? Along with carbon, protein and nucleic acid, lipid is one of the biomolecules. In this section, I give a brief introduction about lipids and its classification. Okay, so lipids, characters of lipids. Lipids are insoluble in water and soluble in organic solvent. We know that, right? Oils and fats don't get easily dissolved in water, but they dissolve in organic solvent. Okay, the another important character or another important point is lipids are not polymers. Okay, unlike other biomolecules like proteins and carbohydrates, lipids are not made up of a single monomer unit. Okay, they are not polymers. Next, into the types of lipids. So there are three types of lipids: simple lipids, conjugated lipids, and derived lipids. Okay, in simple lipids, there is just an alcohol group and a fatty acid group. Okay, in conjugated lipids, we will have additional group along with alcohol and fatty acid, like additional group like phosphate. Okay, phosphate. It can be a sugar. Okay, so that is what a conjugated lipid is. And derived lipids. Derived lipids are lipids derived from these simple lipids and conjugated lipids. Okay, so that's about the types of lipids. So next is fatty acid. Fatty acid have a carboxyl group and a chain of hydrocarbons. Okay, so what is a carboxyl group? COH, right? And attached to this, there will be a chain of hydrocarbons usually represented as this. Okay, in this in this fatty acid, the carboxyl group is polar and the hydrocarbon group are non-polar. Okay, so this polar and non-polar character give the fatty acid the amphipathic nature. Okay, so this COOH is polar, or we can say this COOH is soluble in water, or COOH is hydrophilic, and this hydrocarbon part is hydrophobic, and this give the fatty acid amphipathic nature. Next is saturated and unsaturated fatty acid. In unsaturated fatty acid, there will be a double bond in hydrocarbons. Okay, so that's the difference between saturated and unsaturated fatty acid. Saturated fatty acid, there won't be any double bond. In unsaturated fatty acid, there will be a double bond. Okay, and in this picture, we can see this is saturated fatty acid and this is unsaturated fatty acid because this doesn't have any double bond, and this here we can see a double bond, and this double bond give a king to this fatty acid, or it just we can say the double bond bent the fatty acid. Okay, and due to this bend, okay, we can see here in this picture the unsaturated fatty acid can't pack tightly as this saturated fatty acid, right? Since there is no any double bond and there is no any bend here, the saturated fatty acid can pack tightly, but the unsaturated fatty acids can't pack tightly because of the bends. The we can see the yellow stripes here. These are the kings or the bends due to what? Double bonds. Okay, so due to the double bond, create a bend, and that bend make the unsaturated fatty acid lessly ordered or poorly ordered. Okay, so unsaturated fatty acids are poorly ordered and saturated fatty acids are perfectly ordered. Okay, so next is its state. The saturated fatty acids are solid in state at room temperature. Okay, and unsaturated fatty acids are liquid in room temperature. Like, for example, oils, vegetable oils are liquid, right, in room temperature. At the same time, saturated fatty acid like ghee, vanaspati, those are saturated fatty acids and they are solid in room temperature. And the next point is melting point. Saturated fatty acid have a high melting point and unsaturated fatty acid have a low melting point because of the double bond. Okay, so that's it about the fatty acid. Next is fatty acid nomenclature. So Fatty acids are abbreviated, sometimes abbreviated with such numbers 16 is to 0 or 16 is to 1, 16 is to 2. So what this specifies? This specifies the chain length and number of double bonds. Okay, this specifies the chain length, how many carbons are present, or the chain length specifies. This 16 specifies the chain length. Okay, the 16 specifies the chain length and the number of double bond. Here the number of double bond, this 1 and this 2 specifies the number of double bond. Since this is 16 is to 1, there will be only one double bond in this fatty acid. Okay, and there is a COH group. Okay, so I just drawn the fatty acid like this. That's a COH in front of this. Okay, and here in 16 is to 2, there will be how many double bonds? There will be 2 double bonds. Okay, so this specifies, this abbreviations specifies the number of carbons or the length of the chain and number of double bond. Okay, so along with this 16:1 or 16 is to 1, sometimes we write delta raised to 9 or delta raised to 9 comma 50. Okay, so what this delta raised to 9 specifies? This delta raised to 9 specifies the position of the double bond. Okay, so here the position of the double bond will be between carbon 9 and carbon 10. At the same time, here the position of the double bond between 1 will be between the carbon 9 and carbon 10, and the another one will be between carbon 15 and carbon 16, because here written as 15, so the position of the double bond, second double bond, is carbon 15 and carbon 16. So I have a question here. What will an 18 carbon oleic acid with one double bond abbreviated as? Okay, so there is an 18 carbon and one double bond. So what will be the answer? What will be the answer? So since there is an 18 carbon, it will be 18 is to 1 double bond, right? So 1. So the answer will be 18 is to 1. Okay, I think you guys understand how to nomenclate a fatty acid. Okay, so next, lipid classifications. So this is the main classification. They are mainly classified into storage lipids, structural lipids in membrane, and lipids as signals, cofactors, and pigments. Okay, in storage lipids, the lipids are stored as fat droplets, which we can further utilize as an energy source, like example triglycerols and waxes. And the next is structural lipids in membrane. So they are mainly used for structural purposes like the phospholipid bilayers. Okay, so here example phospholipids obviously, and glycolipids and sphingolipids. And next, last one is lipids as signals, cofactors, and pigments. So there are few lipids which take part in signalings, and they act as pigments which absorb lights. Okay, so we will see each in detail in upcoming slides. Okay, triacid glycerols. So storage lipids, one of the example is triacid glycerol, right? So triacid glycerol, also called as triglyceride and fat, they serve as metabolic fuel. About the structure, they have one glycerol and three fatty acids. So we know the structure of glycerol. Glycerol carry 3 carbon, and all the 3 carbon will have an OH group. So we draw this 3 carbons, and all the 3 carbon will have an OH group. And the first and the third carbon will be balanced out with H2. Okay, so this is glycerol. And about fatty acid, we already learned the structure of fatty acid, right? Fatty acid will have a carboxyl group and a hydrocarbon chain. Now this one fatty acid will form a ester bond with the first carbon of the glycerol. For that, the H from the first carbon and the OH from the carboxyl group will react and release as water. Okay, and the remaining fatty acid will form a bond with oxygen, an ester bond will be formed. So an ester bond within a cell group is formed. So this is how the fatty acid form ester bond with glycerol. Like this, other hydrogen groups of the glycerol will be replaced by fatty acids. Okay, and that's the structure of triglycerol. Here you can see three fatty acids and one glycerol having three ester bonds, and this is the structure of triglycerol. So that's about the structure of triglycerol. And according to the types of fatty acids, the glycerides are divided into two, simple and mixed. So if the three fatty acids are same, then they are simple, and if the three fatty acids are different, then they are mixed. So here in this picture, all the three fatty acids are different, right? So this glyceride is a mixed triglycerol. Okay. Next, the two significant advantages of using triglycerols as stored fuels. So the first one is oxidation of triglycerols yields more than twice as much energy gram for gram as the oxidation of carbohydrate. So this means the energy from fat is more than that of carbohydrate, or the oxidation of fat yields double the energy of carbohydrate. And the second significance is triglycerols are hydrophobic and therefore unhydrated. Okay, this means they don't mix with water and easily break down, and they can be stored for long term, unlike carbohydrate. Okay, next, waxes. So when we say waxes, the first thing came in our mind will be bee wax, right? So not only bees and other insects, some plants also secrete waxes as part of their defense. So this wax are made up of long chain fatty acids and long chain alcohol, and also the melting point is higher than that of fat or triglycerols. Okay, we listed the wax as storage fuels, right? And yes, it is the major storage fuel for planktons. So that's about waxes. So we finished storage lipids, and next we are going to see about structural lipids or membrane lipids. So in membrane lipids are divided into three. One is phospholipid, another is glycolipid, and archibacterial ether lipids. Okay, in this section we are going to concentrate about phospholipids and glycolipids. Okay, so here you can see a structure similar to triglyceride, right? Here also the main difference is number of fatty acid. Okay, third fatty acid is replaced by a group, here a phosphate group, here the monosaccharide or disaccharide group. So depending upon this group, they are divided into phospholipid and glycolipid. If the third group have a phosphate group, then they are called phospholipid. If the third group have a sugar group or carbohydrate group, they are called glycolipids. Okay, so again these phospholipids are divided into two depending upon their backbone. Okay, if the phospholipid have a glycerol backbone, they are called glycerophospholipids, and if phospholipid have a sphingosine backbone, they are called phosphosphingolipids. Okay, and the glycolipids having sphingosine backbone are called glycosphingolipids, and glycolipids having glycerol backbone are called galactolipids. Okay, so next section we are going to see about glycerophospholipids and galactolipids and sphingolipids. Okay, glycerophospholipids. So this is the structure of glycerophospholipid. Here you can see a glycerol and two acyl group linked with ester linkage, right? This entire structure, the glycerol and two acyl group, is called diacylglycerol or DAG. Okay, so along with this DAG, a phosphate group is attached at C3 carbon. So DAG plus a phosphate group is called phosphatidic acid. This entire structure is the backbone of all the glycerophospholipid. Okay, so in glycerophospholipid, mostly all the fatty acid on C1 carbon will be a saturated one, and all the fatty acid in C2 carbon will be an unsaturated one, and the head groups will be attached to phosphate group. Head groups like ethanolamine, choline, serine, glycerol are all attached to this phosphate group. Okay, next we see naming of glycerophospholipids. So the glycerophospholipids are named as derivatives of parent compound, the phosphatidic acid, and according to the polar alcohol in the head group. So if a phosphatidic acid have a ethanolamine head group, then they are called phosphatidyl ethanolamine. And if the polar alcohol group or head group is choline, they are called phosphatidylcholine, and if it is serine, they are called phosphatidylserine, and if it is glycerol, it is called phosphatidylglycerol, and phosphatidylinositol 4,5 bisphosphate if it is a myoinositol 4,5 bisphosphate. Okay, so next, in some cases fatty acids form ether linkage instead of ester linkage with glycerol. Okay, ether linkage instead of ester linkage. Examples are plasminogen and platelet activating factor or PAF. So in both the cases, the head groups are called. This is the picture. This is the picture of plasminogen and this is the picture of platelet activating factor. So in both the picture, the first fatty acid form ether linkage with the first carbon. Okay, so that's it. So plasminogen and platelet activating factor are different from other phospholipid in terms of their ether linkage. Okay, so that's about glycerophospholipids. Galactolipids and sulfolipids are predominant in plant cell, and on evolutionary pressure to conserve phosphate for more critical roles favored plants that meet phosphate-free lipids. So we know that in soil the availability of phosphate is limited, right? So instead to conserve phosphate, the phosphate lipids are abundant in plants. Okay, so here in this structure we can see on third carbon instead of phosphate we have a sugar group, right? This is a galactose, so this is called monogalactose diacylglycerol, and here along with the sugar group we have a sulfate group. Okay, so this is called sulfolipids. So sphingophospholipid is the last structural lipid we are going to discuss. So in sphingophospholipid, there is an 18 carbon compound called sphingosine. Okay, so this 18 carbon compound somewhat resembles glycerol, right? What is a glycerol? Glycerol have a 3 carbon compound and all the 3 carbon will have an OH group, right? But here in sphingosine, the first and third carbon will have an OH group, and the second carbon will have an NH2 group or an amine group, and to this NH2 group an fatty acid will be linked by an amide linkage. Okay, so this fatty acid linked to the sphingosine by an amide linkage. So this structure, the fatty acid and sphingosine, that structure is called ceramide. Okay, so this ceramide is a fixed structure. Okay, only the OH or the H will be replaced by other groups like phosphate and sugar. We discussed this earlier, right? So if the H is replaced by phosphate group, then they are called phosphosphingolipid, and if the H is replaced by sugar, then they are called glycosphingolipids. Okay, one of the example of phosphosphingolipid is sphingomyelins, and they will carry choline or ethanolamine as their head groups. Okay, along with the phosphate group, they will have choline or ethanolamine as their head groups. And examples of glycosphingolipids are of 3 types. So based on the sugar or the carbohydrate they carry, they are of 3 types, and we see that in next slide. Okay, So the nature of the carbohydrate decides what type of sphingolipids they are. Okay, if the glycosphingolipids have simple sugar, then they are called cerebroside. Okay, examples are glucocerebroside and galactoside. And if that carbohydrate group have two or more sugar, then they are called globosides. Okay, so next is ganglioside. This ganglioside will have sugars. Okay, will have sugars. They along with that sugar they will have N-acetylmuramic acid or sialic acid. So no matter whether they have a simple sugar or two or more sugar, if if a glycosphingolipid have a N-acetylmuramic acid or sialic acid, they are called ganglioside. So that was about the structure of lipids. Next, we are going to see few examples of lipids that act as signals, cofactors, and pigments. Okay. So first one is phosphatidyl inositol bisphosphate. So this acts as a signaling factor which take part in calcium release. Okay. When a ligand attaches to a receptor, this will indirectly activate a lipase enzyme. Lipase enzymes are enzymes which cleave lipids. Okay. So here our phosphatidyl inositol bisphosphate will be cleaved into two products. One is IP3, another one is TAG. And this IP3 will go and bind with the channels in endoplasmic reticulum, and this channel get opens and the calcium is released from endoplasmic reticulum into the cell. Okay, so this is how our uh, phosphatidyl inositol bisphosphate act as a signal. Okay, so next example is steroid hormones. Okay, so steroid hormones are derived from sterols and they serve as powerful biological signals. Okay, so examples are testosterone. Testosterone is a male hormone and they are produced from testes. Okay, and next is estradiol. Estradiol is a female hormone and they are produced in ovaries and placenta. And next is corticosterols and aldosterols. These both are synthesized in adrenal gland, and the corticosterol take part in glucose metabolism and aldosterol will take part in salt excretion. Okay, so that's about steroid hormones. Next is vitamin D. About vitamin D is derived from lipids. Okay, not only vitamin D, all the vitamins mentioned here are derived from lipids. So vitamin D is formed from 7-dehydroxycholesterol. Okay, so this 7-dehydroxycholesterol are seen beneath our skin. Okay, when when the sunlight fall on our skin, the 7-dehydroxycholesterol is converted into vitamin D or cholecalciferol, and this cholecalciferol is converted into 25 hydroxycholecalciferol in liver, and again this 25 hydroxycholecalciferol is converted into 125 dihydroxycholecalciferol in liver, and this activated vitamin D will do the calcium metabolism. Okay, so that's about vitamin D. Next is about vitamin A. So vitamin A, when the light falls on vitamin A or retinal, it changes its conformation, and this conformation change send out signals to brain for our vision. Also, vitamin A or the retinoic acid is an important in embryo development. Okay, and the fifth one is vitamin E. So vitamin E, we all know that it is a good antioxidant, and also they have a role in fertility. Okay, the researchers found that mouse having vitamin E deficient diet are sterile, so vitamin E play an important role in our fertility. And next is vitamin K. Vitamin K help in blood clotting. And the last one are ubiquinones and plastoquinones. So they act as electron carriers. Okay, we know that ubiquinones act as electron carriers in mitochondria in electron transport chain, right? Same as ubiquinone, plastoquinone also act as electron carrier in chloroplast. So these are the examples of lipids as signals and cofactors and pigments. So we finish our video here, a topic here. This was all about lipid and its classification, and I hope you guys understand it. and we see you in next videos. Bye.